Good morning, everybody, and welcome to the Friday Informatics PowerTalks series. I'm very happy to introduce today's speaker. I know her well from AMIA and also have served together on the Women in AMIA steering committee. Dr. AJ Uzun is director of the Clinical Bioinformatics program at the Lifespan Academic Medical Center and assistant professor of pathology and laboratory medicine at the Warren Alpert Medical School of Brown University. She completed her bachelor's in chemical engineering at Istanbul Technical University and her master in biological sciences and bioengineering at Sabancı University, also in Istanbul. She completed her Ph.D. in chemical engineering at Northeastern University, focusing on mathematical modeling of drug delivery systems and oral drug delivery. And she completed her postdoctoral work at Brown University, focusing on computational genetics of autism spectrum disorders and bioinformatics. Her current work is focused on translational bioinformatics, focusing on autism spectrum disorder and cancer. And she also has her work in the molecular pathology lab. She has many scientific publications and honors. The name of her talk is Machine Learning and Network Biology Applications in Genomics.
Thank you so much, Amy, for the lovely introduction. Good morning, everyone. It's a great pleasure to talk about my work today. Before I start talking, okay, sorry. Oh, okay, it worked. Sorry about that. So before I talk about my research, today I will talk about two different projects. I would like to give you a brief overview of my clinical work and also my research. So as Amy mentioned, I'm the director of clinical bioinformatics at Lifespan. Lifespan is the largest healthcare system in the state of Rhode Island. We also serve to the southern Massachusetts and northern Connecticut. So I joined Lifespan pathology over 6 years ago, and I started the clinical bioinformatics. So as the clinical bioinformatics team, we basically do the day-to-day next-generation sequencing, NGS data analysis. We build pipelines, we build databases, which is everything for the variant interpretation. We build custom-made analysis tools, and also we build web-based variant interpretation tools. One tool we built and published 2 years ago is VARSTEC. It's online. If you are interested in and working on variants, this is a tool you can basically enter variants information. It will give you information from multiple different online databases. So that's my clinical work. So in addition to my clinical work, I have a research group. I have 11 students, research assistants, medical students, and residents and fellows in my group right now. We focus on several different aspects of translational bioinformatics and machine learning. So we work on machine learning-based predictive models in cancer and autism. We build models to, for example, predict risk, outcomes, or recurrence. We also collaborate with another PI at Brown on a network analysis project in cancer, and I'll talk about that today. We also work on genomic data analysis in autism, and we develop genomic variation detection algorithms. And one of the latest ones is a program we developed to detect insertion deletions from the somatic data. And insertion deletions, I'm not sure if you are familiar with that. Insertion deletions are very hard to detect from the somatic data. And especially in the clinical setting, we have to be very precise about this. So we created INDAR Finder. That's actually another tool or program you can use to detect insertion deletions from the somatic data. And it's online right now, and we are about to publish the paper. Okay, so I'll talk about today two different projects on machine learning and the network analysis, network biology. So the first part of my talk will be about autism spectrum disorders. So autism spectrum disorders is under the umbrella of neurodevelopmental disorders. So as you might know, neurodevelopmental disorders are the disorders of abnormal brain development. So neurodevelopmental disorders are presented as impairments in cognition, communication, behavior, and motor skills, and the genetic architecture, as well as the phenotypic presentation, is very complex and heterogeneous. So again, under the umbrella of neurodevelopmental disorders, we study autism spectrum and also intellectual disability. So autism spectrum disorder is seen in 1 in 44 children, according to CDC data. And there is a sex bias in autism spectrum disorder. It is seen in more males compared to females, and the ratio is 4:1. And again, the presentation is with the social and communication deficits, restricted repetitive behavior, and also we see comorbidities such as epilepsy and ataxia. And the studies have shown that early detection of autism spectrum disorder has several benefits, such as speech therapy, occupational therapy, and the project actually started with this in mind. So autism genetics is again very complex and heterogeneous. There are monogenic disorders presenting autism symptoms, such as Fragile X, Angelman syndrome. We also see common Kaplan-Meier variations. These are also in the guidelines for the diagnosis. We also see de novo mutations and autosomal recessive mutations. So there are several studies using machine learning-based models to predict autism risk. And I just include a couple of them here. In one study, the authors basically looked at exome trios with patients with autism and schizophrenia, and they used machine learning to predict the risk. In another study, they used gene expression data. Using a machine learning algorithm, they again tried to subgroup the patients, and subgrouping is another important question in autism because of the heterogeneity of the presentation and the genetic architecture. So in our project, we built a model, a neural network based model, and my PhD student, Ching Wu, works on that project, and this is a collaboration with Eric Morrow from Brown University. So Ching called this model Separate Translated Autism Risk Neural Network, STAR-NN. The reason is that because we are taking into account the effect of variance from the genetic data separately. So I will call it STAR-NN through my talk. So the data sets we used came from Simons Foundation Powering Autism Research for Knowledge, SPARK. So these were whole exome sequencing data. So the first two data sets, we call it WES1 and 2. Those data sets actually came they were actually released about like 4 years ago. And the first data set had over 27,000 participants, including cases and controls. These were basically the patients and their families. And the ratio of cases and controls are about 50-50. And the SPARK WES2 data had about 16,000 participants. And again, this includes also cases and controls. And we use a combination of these two data sets in our model and for training and testing. And then the third data set came out 2 years after these two, and it had over 27,000 participants, and the data set was used for validation of the model. So when we received the data, we of course filtered it and processed it in order to make it ready for the model. So the first thing we did was quality control. That's the first thing we do in bioinformatics. Always we do quality control. We basically filter out the variants with low quality, and then we combine these two data sets. After that, we basically in the autism, the rare variants are attributed for the risk as risk factors. We basically only extracted the variants with allele frequency less than 1%. So that means these are rare variants. Then in order to make sure that these were real rare variants, we also use nomad data from the non-neural population. We selected the variants with allele frequency less than 1%. So that didn't change the number of variants we got, but we just want to make sure that we have the rare variants. Then we selected the variants based on the effect on the amino acid effect and classified them. In addition to that, we also received SNP microarray data, and we processed this data in the same way, but this time we wanted to see the common variants in the model. That's why we calculated the polygenic risk scores, and this was also included in our model. So the variant types we included in our study were, the first one was the protein truncating variants. So protein truncating variants include nonsense, frameshift, and splicing variants. Nonsense variants, as you might know, are the premature stop codons. And then we also included missense variants. Missense variants were classified based on missense-based Bettness polyphen 2 constraint model, MPC. That's the combination of multiple missense variant effect prediction algorithms. So based on the MPC scores, MPC higher than 2 were classified as Miss A, and then Miss B was between 1 and 2, and then less than 1, Miss C. So if we look at the amino acid effect of those variants, Miss A is probably damaging, Miss B is possibly damaging, and then Miss C would be the rest. Then when we finished filtering the data, then we did feature selection using a tree-based pipeline optimization tool, TPOT. And we selected 1487 gene features. And those included the PTVs, and then missense mutations. So there is a database called Simon's Foundation Autism Research Initiative gene. This database has all the genes which are identified as the autism risk factors. So the way actually this database is created is manually created from the literature based on the variants found in those genes. So right now there are actually over 1,000 genes in the database. It's, I think, 1,000, probably close to 90. And when Chink actually looked at the database, she selected, she got actually 131 genes from Safari genes. And she looked at if the selected gene features were enriched by the Safari genes, and she found actually 115 genes among the selected gene features. And this is actually a significant enrichment. Then we moved to the start and we built the model. So in our model, again, we considered the variant effect separately. So the model, we started the model with the gene features and considering the variant effect. So these 3 networks were basically fed to the model, and then they were merged into one network. And we also included gender and the polygenic risk scores in the model. And then using these networks, we predicted the risk of autism. So the next thing we did was we basically looked at the performance of the model and compared it with the traditional machine learning models. So what we saw actually, our model outperformed all the traditional machine learning models. So as you can see here, we looked at a, I'll show this later, a basic neural network model, and then decision trees, random forest, XGBoost, logistic regression, and SVC. And based on the data, we got accuracy and also ROC UC data, the model actually performed very well. So we also, as I said before, we looked at also a basic neural network model. And in that model, instead of considering the effect of the variance, we only used the aggregated count of the variance. So we basically ignored the effect. And in the same way, we ran the neural network model and then predicted the ASD risk. So the results were actually pretty similar, but the star and N model performed slightly better than the basic neural network model. So we then looked at the model performance based on the gene features we use. So we looked at separately the selected features, the 1487 genes. We looked at only safari genes, the autism risk genes, and then we combined these two. And also we looked at the full gene list, meaning there were over 20,000 genes in the list. So what we saw actually, the model performed in a similar way with the selected features and the combination of selected features and safari genes. The performance was a little low when we used the full gene list. So then we compared the polygenic risk scores with the scores generated by the STAR-NN model. So here you see the polygenic risk scores based on the cases and controls. So cases is the red and the control is the blue. As you can see, there isn't much separation here between cases and controls. Whereas in the model separated the cases and controls very well based on the scores calculated by the model. As I said before, there's this bias in females and males in autism. So we then looked at males and females, the predictions of the autism risk in those cohorts. And what we saw actually, the polygenic risk score, it separated the male cases from the male controls, and the p-value was significant. But when we look at, if you look at the separation in the model, the significance is definitely higher, and it's very obvious. So the model actually predicted the autism risk in the males very well compared to the polygenic risk scores. We did the same type of analysis in the females also, and the polygenic risk scores couldn't separate the female cases from the controls. There was no significance, whereas the model actually successfully separated female cases from the controls. So we then validated the model with a separate, completely unique data set. Although it came from Spark, these were different participants. So as you can see here, this is the model performance on the testing data with the combination of WES 1 and 2 data. And these are the results from the validation data. And we saw basically very similar results in the selected features and selected features with a combination of the safari genes. And the ROC AUC were very similar in the validation data. We then looked at the association of the selected genes with the disorders. So we found that actually there's an enrichment of developmental disorder associated genes in the selected gene features. This is also true for the intellectual disability, pervasive developmental disorder, and also the data showed autism spectrum disorder and autistic disorder. So we then ran gene ontology analysis to see if those selected features, gene features, are involved in any of the biological processes or pathways. So we actually found a good number of pathways and processes, biological processes, as a result of the gene ontology. And for example, positive regulation of potassium ion transport has been mentioned as being associated with autism in the literature. The same for chloride transport and also the potassium ion transmembrane transport activity. There are a couple of them which were mentioned in the literature as being associated with autism. So as a summary, our model STAR-NN show the basically good performance compared to other traditional models, and it could predict the risk of autism in females and males separately very well compared to the polygenic risk scores. And also we found that selected features, gene features, were enriched with autism risk genes, which is actually a great resource for the future autism research. So I'm going to switch gears a little bit to cancer now, and I will talk about the network biology project we have been working on. So that project is a collaboration with Dr. Uzun from Brown University. He's the director of cancer bioinformatics at our the Cancer Center. He also happens to be my husband, so it makes it easy to collaborate with him because I know where he lives, basically. So in this project, we use the network biology approach to certify subtypes in cancer. And today I will talk about, this is a big project. I will talk about only small parts of it in bladder cancer and also breast cancer. So before I start talking about the results, I want to give you a brief overview of the protein-protein interactions and also network biology and how we approach it. So in gene networks, basically, this is the interaction of two genes. So whenever you see these networks, the node is the gene and then edge is the interaction. So we basically look at the degree of a node as based on the number of connections. So if a gene is connected to multiple genes, we call that gene hub gene. So that's the most connected gene, and usually the node will appear bigger in the network. So the complex disorders is one of my, basically it's my focus. So I work on autism and cancer, both of them are complex disorders. And complex disorders are basically the result of interaction of multiple genes and environmental factors. So there isn't one factor we need to take into account, but there are multiple genes and factors in place for the risk of complex disorders. So we basically look at the subnetworks of the disease, which have separate networks, and we look at the phenotype specific networks in the complex disorders. So this is the phenotype specific networks, basically. So this is a slide of a urothelial carcinoma, so showing that cancer is a complex disorder, and not only the presentation but also the genomic factors are very complex and heterogeneous. So in cancer genomics, just very briefly, we take the tumor sample and then we do sequencing. Right. In this world, we do next generation sequencing, and also we look at we do whole genome sequencing, we do whole exome sequencing, we do targeted sequencing, which is very common in the clinical setting because we are still learning about cancer. We basically target only the genes which are diagnostic biomarkers, or there's a target for them. If we try to do like whole exome sequencing or whole genome sequencing, which we can do in the clinic, it takes a long time to basically create the data. Whereas with the targeted sequencing, we target only the mutations, we know if there are any kind of biomarker. We also use RNA sequencing for the gene expression levels, and also in the clinic, we use it to detect fusions, gene fusions. So we take the genomic data, and this is a research project. Of course, I have to talk about clinical work because I'm also on the clinical side. So we take the genomic data and we look at the protein-protein interactions in the data. But the data is a lot. I mean, we have a lot of data, and basically we might be drowned in next generation sequencing. So my collaborator in this project is a cartoonist, and he has a website called Biochemicals. So this is from his website. So showing that we produce so much next generation sequencing data, and we need to spend time to analyze and see patterns in it. So why are we working on protein-protein interactions? Why is network biology important? So Professor Barabasi from Northeastern University works on protein-protein interactions in network biology. And in his papers, he talks about how certain phenotypes have certain protein-protein interactions. So, for example, coronary artery disease have a certain protein-protein interaction map. Asthma has a certain interaction map, and celiac disease, for example, has a certain interaction map. And this is a figure from his paper, and it shows actually different protein-protein interaction maps in different disease or phenotypes. So that's what we are trying to use to stratify cancer subtypes. So interactome is another word I will be using. So interactome is basically the protein-protein map. It's the universe of proteins. So here, for example, I'm showing just the, this is a very bulk picture of the like part of the interactome, and this is also from the Barabasi, one of the Barabasi's papers. It shows actually the different disease and how they are clustering in the interactome. So basically, the similar disease phenotypes, he's saying that in the literature have a strong affinity to interact with each other. So based on this idea, Dr. Uzun and his team developed a tool called Proteinarium. So traditionally, if you want to look at the protein-protein interactions, you will have a gene list, and then you will just use one of the databases, for example, string database, to get the protein-protein interactions. But when this is for one patient. But when you have a cohort of patients, for example, you are studying a cohort of cases and the controls, you will basically do differential gene expression analysis. You will have a final gene list, and now you will look at protein-protein interactions, and you will say that, okay, these genes were differentially expressed, and those the protein interaction is this way, and then basically you will be ignoring the protein-protein interactions specific to the patients. So what they did in the Proteinarium, they took into consideration the protein-protein interactions in each patient or in each individual in the data set. So they basically call it multi-sample protein-protein interaction analysis and visualization tool. So basically you can analyze the data and visualize it at the same time. So what Proteinarium does, you have a gene list. That gene list could come from your RNA sequencing data, your DNA sequencing data, whole exome sequencing data, and then you can basically pick a top n number of genes. And the way we select the top n number of genes is, I'll talk about it later, but we use cophenetic score. So we calculate the similarities in the dendrogram. So what it does, it converts the gene names to the protein names, and then feeds it to the tool. And we call that gene list seed genes. So then it takes the data from each and every sample. Let's say, for example, you have 100 samples and you took top 100 genes. It takes the top 100 genes and now looks at string database, this is the protein-protein interaction database, and calculates the protein-protein interactions for each sample, and then saves that data. And then it maps them into the interactome. So it looks at where those protein-protein interactions are in the interactome. And then it basically finds in the interactome, finds the shortest way using Dijkstra algorithm. So like, if you think about it in biology, you find the shortest way. So that's what that algorithm does. And then it generates the protein-protein networks based on this data for each patient. So then it takes the data, the network data for each, again, individual or patient, and then it calculates the similarities of the networks using the Jaccard index. And based on these similarities, it clusters them and produces this dendrogram. So for example, you have cases in green and then controls in orange. So you can see here, the cases, a subset of the cases are clustered based on their protein-protein interactions. So the next step we usually do, we look at these patients and we visualize their networks. What kind of genes or proteins are interacting is a consensus in that cluster. So you can also check actually one individual patient and then look at the networks, the protein-protein interaction network in that patient using this program. So the output files, the Proteinome gives are the Nubic tree format, so that can be actually visualized using different tools. It gives the gene set and cluster files and also the interaction metrics, which can be visualized using Cytoscape or Gephi. That's what we do. The program actually works in command line, and also it has a graphical user interface. And recently they released the Proteinome 2, which is in beta testing, but you can actually test the program. If you have any questions, you can stop me and we can discuss. I think there's a question in the chat. I'll read that after I finish the slides. So we basically use Proteinome to certify two actually architectures in the urothelial carcinoma. So that project was done by Yiğit Baykara, who is a pathology resident, and Teresa Chow, who is a Brown alum, and she has been working at our labs as a research assistant since last year. So bladder cancer is the 9th most common malignancy worldwide. And in the United States, there are 80,000 new cases and 17,000 deaths every year. And the average age is pretty high. So basically 73% of the patients are older than 65 years of age, and is more prevalent in males, and the risk factors include smoking and chemical exposure. So urothelial carcinoma is the predominant histologic type of bladder cancer. So that's what we studied in this project. So urothelial carcinoma has two architectures, papillary and non-papillary. So papillary actually grows out of the tissue like this way, and whereas non-papillary is more flat. And we actually, in terms of the histology, in the presentation, they can be separated, but in terms of the molecular profile, it's still being studied. So we decided to look at these two architectures to see if there is a distinct separation in terms of the protein-protein interactions. So we use data sets from, we got the data sets from CBOI Portal. CBOI Portal is a database of the Cancer Genome Atlas Program, TCGA data. So it's the user interface. And we got the data, two data sets. One had 133 papillary patients and 273 non-papillary. And then data set 2 had 39 papillary and 88 non-papillary patients in it. So we ran proteinarium in the first data set. And what we saw actually, this is a dendrogram, basically the clusters of the patients with the protein-protein interaction. And we basically looked at the one of the significant clusters. So after we make the dendrogram, we also calculate the significance in those clusters based on the number of cases and controls. So one of the significant clusters gave us this network. So that network A was specific to the papillary patients. And basically, we saw that there were 3 hub genes, RPS27A, UBA52, and WAMP8. Whereas in the non-papillary specific network, there were 4 hub genes, GMB1, RHOA, UBC, and FPR2. And we looked at actually if there were any drugs available for any of those hub genes. So the idea is that you can target those hub genes, and in theory, the targeting should basically take down the whole network and then terminate the tumor. So this actually there will be another collaboration with a PI at Brown to do some bench work. So we saw that there's actually a molecule, a drug used to target GMB, and again we will try to do some bench work to validate this. So the other thing we do actually when we detect these networks specific to the subtype or the architecture, we also calculate the separation between them in the interactome. So there is a score called separation score, which would basically calculate the distance of these networks. I'm showing here these 2 networks, how far they are away from each other in the interactome. So what we found actually based on the separation test, these 2 networks are separated. So basically these papillary and non-papillary architectures show distinct protein-protein interaction networks. So when we also ran the second data set, and we didn't get significant results from the non-papillary group, but the papillary group actually gave us this very significant network. And here also the hub genes were CCDC22, NFC4, and UBR4. And the papillary specific networks in dataset 1 and 2 also shared these 6 genes. So the next project I will talk about, this is on breast cancer. So in this project, a former pathology fellow, Sean Hecking, worked on. He's right now an assistant professor at the University of Toronto. And again, Yeet and Trace also worked on this project. So in addition to looking at the very basic subtypes of breast cancer, ductal and lobular, we also studied mismatch repair protein deficiency. So MMR. So MMR is not really studied well in breast cancer, whereas it's in the guidelines for the colon and endometrium cancer. So we basically check MMR status for this type of tumors. So in the world, breast cancer is very common. Basically, one woman diagnosed with breast cancer every 9 seconds. That's a very high number. And the mismatch repair protein, MMR, deficient breast cancer is only 2 to 3% of the breast cancers. What we saw in this study, which I won't show that, but we saw that most of those patients are triple negative and ductal. They have ductal carcinoma. So we wondered actually if MMR deficient breast cancer will show a distinct protein-protein interaction network compared to the MMR intake. So the data set we used is again from CBOY portal, the TCGA data. So in this data set, there were 29 MMR deficient patients and 965 MMR intake patients. Again, the 2-3% is actually also presented in this data set. So what we saw in this data set, we basically analyzed it using Proteinarium, and there were actually the subgroups, the MMR deficient and the MMR intact groups were very significantly, I mean, obviously separated. So this is the MMR intact group, which shows actually the hop genes GRB2, AP2B, DING1I2, EPS15L1, and CDC42. Whereas the MMR deficient network showed heavily histone proteins. So it's basically there are histone proteins from histone H2B family, and there are additional genes CEMPL and CCMB1. There are actually histone targeting drugs available. So one of the things in this study, we are about to publish it, we actually made recommendations for checking the MMR status and considering those drugs for those patients. So again, this is these are the small parts of a big project. So the big project, we call it Atlas of Protein-Protein Interactions in Cancer, APIC. So APIC actually will be a website, an interactive website showing the protein-protein interaction maps in several different tumor types and the subtypes. So as you can see here, there's actually, this is a list of the cancer types we have included in APIC, but we are also working on including other cancer types and subtypes. So you will be able to basically, this will be the opening page. You will be able to go to the page and click on, for example, the colon, and then it will bring up the whatever subtypes we looked at, and you will be able to visualize the protein-protein interactions in a 3D way. And you will be able to do the same, for example, for lung cancer, for prostate, for thyroid, and other cancer types. So as a summary, in those small projects, we showed that the papillary and non-papillary urothelial carcinomas present distinct protein-protein interaction networks. Also, Proteinarium showed that the MMR deficient breast cancer has a unique protein-protein interaction with heavily connected histone genes, and also histone proteins, and also we can basically use this information for drug repurposing in the future, and we are hoping to do some experimental studies to validate these findings. So with that, I would like to thank to our collaborators. We have been collaborating with pathologists and oncologists and basic scientists from Brown, outside Brown, from Yale, there are people actually who have not been listed here. And these are the people who made these projects happen. These are my lab members. And if you would like to check out my lab, this is the website. We also organize a journal club, a monthly journal club. It's on machine learning, it's specific to machine learning and genomic applications. If you would like to join our website actually has the information on the journal club, how to register, and you can join. We mostly have guest speakers who are the authors of the papers we would like to discuss in the journal club. So I would like to thank to the funding agencies, also most importantly families from Safari. Reichardt is the registry for the autistic patients in Rhode Island. I'm also involved in the program for developmental disorders genetic research program, which is the Dr. Marrow, my collaborator, is the head of it, so I've been involved in that too. So without the families, we wouldn't be able to do any of this work. This is part of my group, and also Dr. Uzun's group. This picture was taken at the beautiful park in Rhode Island in May. It was a very windy day, you can tell. So we are hoping to take more pictures. After COVID, this was our first in-person meeting as a group. And I took this picture in Providence. This is near where I work, near the medical school. It's actually, Providence is a beautiful city. With that, I would like to thank you all. I'll be very happy to answer your questions. Thank you.
Well, thank you, Dr. Uzun, for the excellent presentation. There is one question in the chat, and feel free to enter your questions and comments in the chat, or you can also unmute yourself.
Yes, the web platform is definitely available for you. If you Google Proteinarium 2, you can actually use it. And also Proteinarium 1 is available on another web page. If you Google them, you will find also the paper is available. It's on the PubMed. So paper has the link to the Proteinarium 1 website, and Proteinarium 2 is available as a separate website.
I have a question.
Yeah.
How do you define the hub genes? You have multiple genes in that network. The other question I have is your protein-protein interaction database based on data not related to your phenotype of interest. So how do you define the case and control that was generated by data that's not really related to your phenotype?
Okay, these are, yeah. So first of all, these are great questions. The hub genes basically are the most connected genes. So we basically sort them based on the interactions they have, how many genes they're interacting. For, like I showed you, for example, top 3, 4. These genes have like 5, 6 gene interactions. So the other, the answer to your other question is, so we do this in a blind way to discover new protein interactions in the phenotype. We basically take the data from the string database, and we use only the experimentally validated data. So basically, we cluster the patients or all the cases and controls based on their network protein-protein interaction networks, and those clusters, we know which ones are basically the patients, which ones are the cases. So the clusters of the networks, if they are specific to the cases only, so let's say, for example, in a cluster, we have 10 patients, but 1 or 0 control. So that means that network, the protein-protein interaction network, is specific to those cases. I don't know, is that clear?
Yeah, I was trying to, you mentioned the string database.
Mm-hmm.
So you are using cancer. If the string database is not related to cancer, how good is your, clustering? If the database was based on cancer, then you could be able to cluster your patient based on cancer database. But the string database probably generated post-mortal or any other healthy individual, which may not be reflective of in the disease status.
Mm-hmm. Yeah, so again, so basically, if you look at the only cancer-specific protein-protein interactions, you are basically focusing on only those. But we are trying to discover other protein-protein interactions, not just, discovered before. I don't know if it makes sense. So we are trying to, our approach is more general, but we are trying to specify the phenotype-specific networks. So basically, the drugs, we try to do drug repurposing. Some of the drugs are used for example, for eczema, but there's a potential for the drug to be used as a target for a subtype of a tumor.
Thank you. I can ask one more question about the star and neural network versus the basic. So in your neural network model, can you give weight for different type of markers? Like, there's a functional variant. All these are assumed to be equally important. Is there a way to give weights based on the functionality, or can you also incorporate other than molecular marker, for example, clinical or other environmental factors?
You basically described our next steps. It's in progress. So first of all, we are trying to incorporate the clinical phenotypic clinical data, which is very heterogeneous. So we have been studying it and trying to subgroup the phenotypic data, and the giving the weights is also in progress. I just show it. Yeah, you basically describe what's going on right now. My PhD student is working on those steps.
Now, as a follow-up to the chat, the gene set 1 and gene set 2 need to be entered. However, I just want to see any interaction within a single gene set. Can it be done?
Oh, yes, you can do it. So one of the things we do, we just look at only one gene set to see the clusters. If there are any clusters in the data. So you basically do it in a blind way. And if you want to, for example, you have a specific subtype or any type of tumor or any other disorder, and you want to see if there are other subtypes which have not been defined, you can use that approach actually to define those subtypes. Yeah, definitely. So this may be a technical question. For the deep neural network model you presented, did you find adding more hidden layers could improve ROC, or number of hidden layers doesn't matter much for that case? Thank you. Yeah, yes, Ching tried that. She tried to add more hidden layers, but it didn't help us. So we used 0 to 2 layers. She tried higher, but yeah, it didn't help, unfortunately. Which set of interactions from string is included in Proteinarium? We only included the experimental validated interactions. I don't know if that's your question.
Thanks everybody for all the great questions and comments. Okay, that was the question.
Okay.
Any other comments or questions? We have a few more minutes if people have some burning questions.
Yeah, I can have one. On your ROC curve, which you compare star neural network with traditional machine learning, at least from the ROC curve, I don't see much difference. They're just line up all together, and it's not really that high. I don't know what sample size you have, because the sample size is not clear here, but it's around 7.7 or around that. So what is the benefit of doing the new one?
So so I can go back actually. Let me see. Okay. Oops. Let's see if I can show you. Yeah.
Yeah.
So you are referring to this, right?
I think so, yeah.
Yeah. So yeah, I know there's a slight difference, but we are trying to improve it. So this is basically a start. So our one of the things we are trying to do, we are trying to add copy number variation data. These are very common in autism, and they're important. So that's one of the things we are trying to do. And we are hoping that this will actually increase our accuracy, and also the clinical data will hopefully, some of the features will increase it. So this is just the start. I know it's slightly different, and but it's different from if you use like decision tree or random forest, it's pretty high compared to those. But I agree that the basic neural network model compared, if you compare with the star NN, yeah, there's a much difference. But we are trying to improve the model by adding more features to it. Yeah, no, that's a great question. Any other questions?
It's Jim Semino here.
Hi.
Director of the Informatics Institute. I just wanted to say thank you for contributing to our seminar. We had a great turnout. We got great discussion, and I really appreciate your time, especially as an alumnus of Brown University. Nice to see folks from Providence. And yes, Providence has become a beautiful city. It was not when I was there, but it's really gone through some remarkable changes. So
Yeah, I don't know.
Thank you very much.
Oh, thank you very much for inviting. I don't know how long you haven't been to Providence, but it's completely different. I've been here since 2010, especially the area where medical school is. There's a new building. It's amazing. I think you should come visit us.
Yeah, I was back there about 5 years ago, and it was, it's remarkable.
Yeah.
But, and say hi to my former trainees, Liz Chen and Neil Sarkar.
Oh, I have a meeting with them at 3 today. I will. I will. Definitely.
Yeah. Oh, I talked to Jim today. Any final comments or questions? We're just about out of time. And you can always contact Dr. Uzun, and we'll know how to reach her at by email. All right, if none, then thank you very much for attending and your wonderful questions. And thank you, Dr. Uzun, for a wonderful presentation. And we have a great weekend. We hope to see you at the next Power Talk. Goodbye.
Thank you. Thank you so much. Thank you. It was a pleasure. Thank you. Have a great weekend. Thanks.